Okay, let's get started. Hello, everyone, and you're very welcome to our live webinar this evening or this afternoon, depending on what part of the world you're in. And I'm sure we've people from all over the world joining us. Our webinar this evening or this lunchtime is how bimodal neuromodulation can soothe tinnitus symptoms. It's about the recent research paper that was published in Scientific Reports, a scientific journal in the Nature family, titled Different Bimodal Neuromodulation Settings Reduce Tinnitus Symptoms in a Large Randomized Trial. My name is Neil Doyle. I'm the Marketing Director at Neuromod Devices, and my job today is to just host and facilitate the discussion with our esteemed guests. There is an interactive Q&A function. The session will last about an hour's time. We have set aside time at the end to take your questions in an interactive fashion, but please feel free to use it up until that point. And let's get started. I'm looking forward to the discussion, and I'm sure you all are as well. So that's enough for me for now. Might ask our speakers to introduce themselves. I'm sure a few people on the webinar know who our speakers are, but for any of those that don't, Ross, I might ask you to introduce yourself first.
Yeah, thanks, Neil. I'm Ross O'Neill. I'm the founder and CEO of Neuromod Devices. I come from a background in biomedical engineering. I have a PhD in the area of neuromodulation, and more specifically bimodal neuromodulation. And I also have an MBA from Trinity College.
Brilliant, thanks, Ross. And Hubert, I might come to you and ask you to introduce yourself and give some background.
Sure, and Neil, thanks for moderating this. Hey everyone, my name is Hubert Lim. I'm professor at the University of Minnesota in the Department of Otolaryngology and also in biomedical engineering, also serving as the Chief Scientific Officer of Neuromod Devices. My lab at the university, spans various hearing technologies, and also how you stimulate or activate the nervous system and the brain for driving changes in the brain for treating things like tinnitus, pain, and also immunomodulation. And in my lab, I've been working on bimodal neuromodulation for some time, and it just worked out well that I could come to the company Neuromod Devices since 2017. Even moved here for a bit to help push forward bimodal stimulation through Neuromod Devices.
Very good, thank you, Hubert. And Ross, I'm certainly looking forward to the discussion today, and I hope I can keep up with you. I do not have such a scientific background or research background, but Hubert, it probably a point of important information for our audience is you're an author actually of the research paper that was published. I think, in fact, the corresponding the author, if I'm not mistaken.
That's correct, and I am serving as the Chief Scientific Officer, as I mentioned. So I am overseeing the clinical and scientific activities in the company, and one of those items is these clinical trials. So TENT-A2. As well as TENT-A1. So that's the reason for me overseeing the study and also being the last and corresponding author of the paper. I'm still closely tied as well to the University of Minnesota. There's complementary research going on there. And so there's opportunities for collaborative efforts as well from both sides. And there's ongoing collaborative efforts as well as we speak.
Great. Let's dive in, I guess, into our topic. So I suppose tinnitus probably needs no introduction to our audience. It's obviously a significant global healthcare need, and depending on which research you read, approximately 10 to 15% of the adult population globally may experience it at some point in their lifetime. And there's obviously a treatment ecosystem or numerous interventions that can be used for tinnitus, but I suppose this paper and the trial was investigating bimodal neuromodulation specifically. And before we get into the real scientific detail and research detail of what that is, it might be helpful to get an overview of neuromodulation generally, and bimodal specifically. And Ross, maybe I can come to you for just a quick overview of what neuromodulation is or isn't, and what it hopes to achieve.
Yeah, so neuromodulation is the use of electrical stimulation to modulate or alter activity in the human nervous system, and that's for therapeutic benefit. Bimodal neuromodulation pairs, and specifically with our technology, pairs sound therapy with electrical stimulation. This is an approach that is well known in neuroscience, called paired associative stimulation, and it dates, its origins date right back to Pavlov and the dogs. And it basically involves presenting two forms of stimulation, as I said, sound with electrical stimulation, and you present that to the brain over and over and over again to drive a process called neuroplasticity, which is how we wire our hardwire effects into the brain. So that's what we're attempting to do with our technology, is essentially to drive long-lasting therapeutic benefit and kind of combat the underlying neuropathologies of the tinnitus condition.
Very good. And I think the assumption there is that, tinnitus is a neurological condition, and that's why this kind of therapeutic intervention, I suppose, is interesting and has been researched with the work that you've been doing. So from a bimodal perspective, I suppose it's two treatment stimuli paired at the same time to achieve the kind of effect of the nerve activity. Is that a fair simplification, I suppose?
I think that's correct. I think that, as you said, it's an incredibly common condition. It's one of the largest unmet global healthcare needs, 1 in 8 adults. The standards of care are either hearing aids or CBT, and hearing aids are not a great fit for tinnitus patients for a number of reasons. They're recommended for hearing loss, not for tinnitus, and a lot of younger patients, they have no measurable hearing loss, a lot of younger tinnitus patients, so they're not recommended for hearing aids straight away. Patients that are in kind of middle-aged typically don't opt for hearing aids because they see tinnitus as their primary problem, and also they're not ready to wear hearing aids yet. And then older patients who do use hearing aids and have tinnitus, they report that when they take the hearing aids off at night, that their tinnitus spikes, and it gives them difficulty in sleeping. And that all the literature says that hearing aids and sound therapies can be effective while in use. They help mask the tinnitus, but they don't provide long-lasting relief. So one of our motivations for this was to provide, to kind of turbo boost sound therapy, if you like, to drive long-lasting effects. And that is one of the reasons that we came up with bimodal, as I said, paired associative stimulation, pairing sound therapy with electrical stimulation to drive neuroplastic changes and to try drive that long-lasting relief that patients are looking for, that unfortunately hearing aids and sound therapies are not providing.
Very good. Okay, I'm sure we'll definitely unpack that in more detail, as we go through the granularity of the trial design and the results. Hubert, I might just get your perspective here then, your scientific perspective on, so why is bimodal potentially more effective than bimodal neuromodulation, potentially more effective than neuromodulation, or good old single neuromodulation, if you will?
Sure. I think you have to put it into context as well. I wouldn't say necessarily bimodal is better than, unimodal neuromodulation. Depends on situation. We all know, situations like Parkinson's, tremors, they do neuromodulation and stimulate and actually works quite well, in certain subset of patients. It's not to say if you were to stimulate multiple pathways and figure out the circuitry that you couldn't improve it, to be determined. But for our purposes, for tinnitus or tinnitus, however you prefer to say it, our mechanism of action is that there are cells in the brain that are coding the tinnitus, and you have some options where you can interact and shut down those tinnitus cells, and you can try to reduce the activity of them, or the abnormal activity of them. And sound actually can be effective to interact, as Ross was mentioning, and suppress them maybe for a short period of time residually. And that can be, you could think of sound stimulation as a form of neuromodulation, right? Because it's still modulating the brain and the cells. But bimodal neuromodulation, as Ross already mentioned, alluded to, was that, the way the brain works, it has to work in patterns, right? And we know that, when there's synergy or synchronous things happening, the brain takes notice of it and can also change its coding, to reinforce that. And if you think about the old school Pavlovian dog, where the dog could care less about the bell, or whatever, stimulus is provided to it, but then you pair it with food, then the brain is lighting up and coding for that bell, even when the food isn't present. So that's been known for over 100 years, right? So all we're doing here is now combining the sound with electrical stimulation of the tongue being, you could think of like a reinforcer, right? That does project, in my lab, we've shown this too, does project and modulate cells in the auditory system where tinnitus is coded. And then you do it together and it tells the brain, whatever the sound's doing, reinforce it, make it more enhanced, cause a long-term effect and stick around, right? So this is the way to view. So in that sense, you could say it's better than unimodal because it's driving a much stronger reinforcement, effect into the brain. And then of course, we'll get into this later if people have questions. We play lots of different stimuli because we want the brain not to focus on just that bell or that one stimulus. We want the brain to be, providing its attention or awareness to many different stimuli other than the tinnitus, and then reinforcing that state. That's the idea behind it.
Brilliant. Thank you so much, Hubert, for that. So we might just move on then specifically to the Lenire device, which was the device trialed in the clinical trial and is behind the research paper, and maybe just give a little bit of context then on how specifically the bimodal neuromodulation is provided before we get into by the Lenire device, before we get into the specifics of the trial discussion and design. So Hubert, I might just following on from what you said initially, Ross, was sound stimulation combined with mild electrical pulses to the tongue. So I guess at a very basic level, how does the device do this?
Yeah, so the device has three components. It has sound therapies delivered via headphones. These are just kind of consumer headphones that are Bluetooth, buy them off the shelf. And the paired simulation that's delivered transmucosally to the tongue is via a proprietary piece of technology, an intraoral electrode that we call the tongue tip. And it basically goes in the mouth. It's a non-invasive stimulation, so it goes in the mouth like a lollipop. You close the mouth and delivers mild electrical stimulation to the nerves at the end of the tongue, and that's why we call it the tongue tip. Then the sounds that you hear in the headphones and the stimulation that you get on the tongue are paired, and this is controlled by a handheld device called the controller. Where the levels of intensity and the volume of the auditory stimulation can be adjusted by the user. The patient uses the device, for 30 to 60 minutes per day. And as I said, the idea is to pair these two stimuli again and again. So over a treatment session of 30 to 60 minutes, a patient gets thousands or hundreds of thousands of these individual stimuli. And then over 12 weeks of treatment, they will get millions of these stimuli. And over time, that drives these neuroplastic processes that drive that kind of hardwire that therapeutic effect into the brain.
Very good. And that's so just maybe two quick follow-up questions. So the first is, it's intended for use from home, that the patient does it from home for the 30 or 60-minute daily treatment slot, right? Yeah. And then when you say neuroplasticity, maybe for neuroplastic effects, just for people in the audience maybe who aren't familiar with that terminology or concept, that is the ability for the brain to kind of rewire itself over time, I believe. So for example, if you are learning a piano, it's maybe you're very clunky with your hands for your first 2 or 3 weeks while you learn the keys, but in a year later, it's natural, or maybe like driving manual or driving with a manual shift car. the brain's ability to rewire itself, right? So that's
correct,
what we're trying to achieve. Okay, cool.
Yeah.
And
so they say it takes 90 days to learn a new skill. So, you know, whether you're, as you say, if you're learning piano, juggling, whatever, it takes 90 days. And that actually is that kind of magic neuroplastic window. It's the kind of amount of time that the brain takes to hardwire these new habits into the brain. So that's around about the same. You know, we recommend 12 weeks of usage. So that's that magic kind of neuroplastic 90-day window.
Got it. Very good. I might just come to you, Hubert, to follow up on something you mentioned previously about the tongue, which is being stimulated here. So why the tongue, as well as, you know, sound stimulation? Because I think for people with tinnitus, it's maybe a little bit obvious or expected that, you know, you play some sound through ears to potentially help with tinnitus. So why the tongue?
Yeah, so a little bit of background there. You know, it's about 30 plus years, 40 years of research has shown that there are converging pathways, right? So the auditory system, you have sound coming in, goes into cells in the brain that are coding for sound. And then you also have in the body cells that are coding for the body, somatosensory pathway or tactile pathway. But they discovered, you know, many studies now that these two pathways do converge, and they converge in the auditory system, they converge in the somatosensory system. But you could think of, if the cells are in the auditory system, right, and they're abnormal, then you have your sound coming in, you have your somatosensory pathways, but they do converge in those auditory brain regions. Now, that's stuff we already knew, but in my lab, we want to see if you do stimulate different body regions, the tongue, the ear, the face, the neck, the body. I mean, we stimulated all over in animal experiments, and we combined it with sounds, different kinds of sounds, tones, noise, different types of patterns, delays. What was interesting, you know, I figured you would see that electrically stimulating the ear and sound would have the strongest plasticity effects, and we did see strong effects there. But one that actually popped out that was also a strong effect on this plasticity was tongue stimulation. And I actually published that paper in my lab in 2015. And in the conclusion, I said tongue is a you know, important region that can drive strong plasticity. And to be honest, that's actually how Ross and myself got together, because they had read that paper and they were obviously working on tongue stimulation with sound. And I'll give credit to MME because she had found that paper and presented it to the group. But it just worked out synergistically that they, you know, Neuromod was working on tongue and sound. We had discovered in animals that tongue was a good candidate. So, you know, from a neuroscience anatomical point of view, you know, stimulating two regions, somatosensory, auditory, makes sense. As to why tongue, it just happens to be from animal experiments that tongue was a strong driver of plasticity.
Good. So it's an effective place to stimulate to get the effect of trying to manage the tinnitus or manage the change in the brain to reduce the tinnitus symptoms. Then.
Exactly.
Yeah, so there are a couple of pieces of literature pointing that direction. You know, there's pointing towards trigeminal nerve, you know, inhibiting some of the neuropathologies that were believed to underlie tinnitus. So I think a lot. The reason we chose the tongue is that it's one of the most innervated parts of the body. It doesn't have a mucosal layer. It's very easy to stimulate. You can stimulate at very low levels of electricity, you know, so very safely. It has a naturally recurring conducting electrolyte, which is saliva. So it made sense on multiple levels. And as Hubert said, he had published in his lab that it actually was, from an electrophysiological perspective, it was one of the best places to stimulate. So it was quite serendipitous that our own research here in Ireland and Hubert's research in Minnesota kind of both converged on the same point. And that's when, you know, we got together and started collaborating on this technology.
Very good. And I suppose it's probably important to note that, so the tongue, it's not just a good place to stimulate, but it's practical and comfortable and usable from a patient perspective. I suppose it's potentially great to have some research that says, you know, this can work, but is it accessible for patients to be able to actually use, you know, from the comfort of home, etc., or so on.
And actually, Neil, just to add to that, about your comment about comfortable, you know, when we say tongue stimulation and the device in the mouth and the oral area, you know, we do hear back comments, oh, geez, like that seems like it would be inconvenient or uncomfortable and so forth. But it's actually surprising when you use the device. You know, I think if you ask people about a lollipop in your mouth, you know, I'll have a lollipop in my mouth, you know, with my kids, and you know, we won't even realize it's there, right? It's kind of like that. And when you look at, we'll get to the data, but you know, it is interesting when you see the high compliance rate, right? We're talking 80, 90%, and even people who have you know, thought positively and satisfied, even recommend the devices 80, 90%. We'll get to that, but you know, it just shows that it actually is quite comfortable. It just maybe the first few times you get used to it.
Brilliant. That's good. Thank you, Hubert. So maybe just one final point on this before we move on to the research more generally. We're going to speak in the research about, or we're going to speak as we discuss the research about this concept of changing stimuli or changing the treatment that the participants received. How is this device able to change the stimuli that the participants are able to receive? Ross, maybe I come to you for that one.
Yeah, so we can program the device with, you know, any form of stimulation, you know, multiple, as we call them, parameter sets. So we, as Hubert said, you know, one of the lines of inquiry in our research has been, you know, the effects of different stimulation parameters on therapeutic effects. So both in the sound side of the bimodal stimulation, but also on the electrical tongue stimulation. So the device can be programmed to you know, deliver any of these parameter sets. So typically what happens is that either the healthcare professional, whether it be an audiologist or ENT, or in the case of a clinical trial, a research nurse or a clinical researcher, they can program the device through a user interface on a computer to deliver any one of these parameter sets. So they essentially just plug in the device, program it, and then give it back to the patient who uses it at home. Then, as I said, 30 to 60 minutes per day for at least, you know, 6 weeks before they see their healthcare professional again.
Very good. Okay. Thank you. Let's dive into some more discussion about the research. So the TENT-A2 clinical trial, which was the trial this most recent research paper was written about, is, you know, not the first large-scale trial that took place. That was TENT-A1, and there was, I suppose, previously safety and feasibility studies and pilot study. You know, and this is probably a pretty typical research journey when bringing a treatment intervention into market for any condition, I suppose, really. But why would you look to carry out a second large-scale trial, Ross? You know, after TENT-A1, why do it again for our audience?
Yeah, so the TENT-A1 and TENT-A2 studies were what's called parameter optimization and patient subtyping studies. So we wanted, as I said, we were looking at what differential effects different stimulation parameters drive in patient groups, but we were interested to see, you know, what differential effects they drive in the total tinnitus population and also in different tinnitus subtypes. So there are a number of different tinnitus subtypes, whether, you know, it's hyperacusis and tinnitus, somatic tinnitus, etc. So we wanted to see, you know, moving towards a kind of a personalized treatment, would we see differential effects in, you know, if we provided different parameters to those different groups, or potentially in the whole tinnitus population. So in the TENT-A1 study, we did that. We compared 3 different parameter sets, and what we saw was, well, first of all, you know, which was very positive, we saw that we delivered a, you know, clinically significant improvement within the first 6 weeks of treatment for the whole population. But probably one of the most notable points was then the treatment effect plateaued out at the 6-week point. So we believe that was due to a phenomenon known as neural habituation. So this is when the brain or the human nervous system, it's essentially habituates or gets used to non-repetitive or non-varying stimuli. So repetitive stimulation. It's the reason why you can't feel your socks after you put them on for a certain amount of time. Your body just adapts, right? So we wanted to see with the TENT-A2 study, number one, if we could replicate the results of the TENT-A1 study, could we deliver a clinically significant improvement in the majority of the tinnitus patients for a second time? But then also we wanted to see if we perhaps change stimulation at the 6-week point, might we overcome this habituation phenomenon that we were seeing, which was resulting in a plateauing of therapeutic effect. So what we did in the TENT-A2 study was patients got a certain parameter set for the first 6 weeks. And then when they come back to the hospital for their 6-week visit, as I said, the clinical researcher took the device and reprogrammed it with a different stimulation set for the second 6 weeks. So we were delighted to see that actually did work. So rather than plateauing out, the patients in the second 6 weeks had further improvement. So it seems that this approach of stimulation cycling overcomes this neural habituation and can lead to what we're hoping is ever improving therapeutic outcomes. Now we're going to continue to investigate that to see maybe if you continuously keep changing up so that the nervous system doesn't get used to it and just start to adapt to the stimulation. So that was the overarching motivation for the TENT-A2 study, but also we wanted to further investigate, continue our investigation around stimulation parameters and investigating if they do drive differential effects, what parameters are absolutely critical to the therapeutic effect, what aren't so much, and also to try to flush out if we could get a more targeted treatment for some of these patient subgroups.
Very good. Brilliant. Okay, so let's unpack it in even more detail. We will also touch later on the real world analysis that was carried out too. So if there's any questions on that, we'll cover that. So just I suppose the high level detail about the trial, it commenced in 2018. There was 191 enrolled participants, which is I suppose a relatively significant size. So we can call it large scale, I suppose. It was blinded and randomized and carried out in St. James's Hospital in Dublin. So we had over 5,500 people, Hubert, I guess, initially, raise their hand to be involved and participate in the trial. How do you get from that number of people who are interested down to the 191 kind of enrolled participants who actually, participate with treatment and the follow-ups?
Sure. And just so everyone knows, I think one of these slides, they'll have a link to our protocol paper and also our results paper. In there, we do provide the criteria, so inclusion, exclusion criteria, and some reasons, in the paper. But the key thing is, we try to keep it as broad as possible. Now, when you're designing clinical trials, you do need to consider two things. One is, it's a clinical trial and you're trying to investigate the safety and efficacy, you want to minimize additional risk situations. So for example, in our study, we didn't include those who may have seizures or who may have implantable devices in the brain or in the head because there might be interactions. These are things that, we have to consider. So there is some safety aspects to consider. But the other thing about clinical trials is, we do need to constrain it to a certain population. I'm not saying it has to be too narrow, but some, intention of what we think we're going to treat. And so for us, we're looking at things that are changing in the brain, and that may, we view it as subjective tinnitus. Now, obviously, there's not a clear, easy way to measure subjective or objective, but we did exclude those who have objective. For example, it might be pulsatile tinnitus and synchronized to vascular pulsations. There may be other reasons, that are mechanically driving the tinnitus percept. But other than those kind of conditions, we did try to keep the inclusion exclusion quite broad, right? Inclusion criteria, so it's more generalizable to the larger population. But when you go through many of those factors, I will say, actually, let me restate one thing, is that there was some arbitrary criteria that are based on some, neuroscience aspects, which is in our first TENT-A1 study, we also did 3 months to 5 years as the tinnitus duration. There is evidence in the neuroscience field that over time things get more settled in the brain. And so we wanted to recruit those who could enable more plasticity. We found great results in TENT-A1, so we broadened that up to 10 years, so 3 months to 10 years. So that is a window of inclusion that we had. And also, other factors like adults, we kept it to 18 years and over. If there's other treatments, ongoing treatments and things like that, to not confound. Because, again, it's a clinical trial, so we have to have some systematic way to evaluate. So these are some things that inherently go into the design. But with all that together, that's basically how you end up going from these high number of individuals. And then, of course, we screen them down and we take them as they come in a randomized order to allocate to the different arms. And that's how we end up getting our 191. It's not to say that, I just want to clarify, it's not to say that there's 5,500 minus 191 that weren't eligible. That's how many came in, but we had to then do our screening process. And then we went in order as they came in to then randomize, right? So many of those individuals probably would have been eligible still. We just didn't have enough slots. So I want to make that clear.
Yeah, I don't know if the technical term will be a very large scale blind and randomized trial, but I suppose you have to appreciate the running a trial for 5,000 participants is the amount of time and hospital hours and stuff that might take to achieve that. But maybe just one follow-up question then on the criteria for who was involved, because this is a trial. Is that to say that maybe there was some things that excluded certain people from participating in the trial that maybe in the real world they could still use the device from a healthcare practitioner that provides it rather than in the trial setting?
I can answer that. So there were some situations that were more practical or logistical related. For example, we have a device and we can play sounds out of them. After certain sound pressure level, the headphones can become distorted, or they don't have enough power to get the sound out, right? So we did have some limits as to kind of the amount of hearing loss bilaterally. That's an example. We also had a cutoff. If people are familiar with tinnitus assessments, one of the gold standards is tinnitus handicap inventory, and the score there goes from 0 to 100. 0 meaning no issue, and then 100 being the worst scale, for that questionnaire. They had to have a 38 score, 38 or higher. They had to be severe enough. But these things we did for the study purposes, for logistical reasons, and some other factors. But of course, that's the clinical trial. It's not to say it won't work in individuals who may have more hearing loss or unilateral hearing loss, right? Or deafness on one ear, severe hearing loss, profound hearing loss, or that they have milder tinnitus symptoms. But it's just the way the study was designed to have some kind of structure and consistency across. So there are a couple inclusion-exclusion criteria like that. But those are things that in the real world, we're observing to see how well they work in those individuals outside of those criteria.
Okay, very good. I think in terms of the objectives of the trial here, I think Ross articulated those previously about why to carry out a second large scale trial after the first one. But maybe just one more general question would be, there's a significant difference here in terms of the numbers of people in arm 1 and arm 2 versus arm 3 and arm 4, Hubert, and why would that be the case, I suppose, when I guess designing the trial or allocating people to these arms. And maybe for people a little bit unfamiliar with clinical research, an arm is essentially a group of participants who are receiving a certain type of treatment. Is that a correct statement?
Yeah, so a little bit of background on clinical trials. You know, when you design clinical trials, you have to make sure that we say the study is powered, meaning there's enough individuals and designed in a way that you could show that there is a meaningful or statistically significant change or improvement. And so you need a certain number of participants for a given condition, right? And that is all determined. There's some math, statistics behind that, and based on some preliminary data, you can figure what those numbers are, for the most part. And also, when you design a study, you need some comparison, whether it's before and after treatment or one treatment version to a control or another treatment. So a lot of things you have to consider. Now, in this study, as Ross already mentioned, there were a few objectives. One is we want to see, by changing the stimulation parameter over time, can you get greater improvements? So we had to kind of a priori, right, pre-design that into the study because, again, you don't want to just post hoc kind of cherry pick results. You want to make sure that you actually had predictions, and then those are the predictions that you're testing, so that builds the clinical confidence evidence for it, right? The other thing is that, as Ross mentioned, we are trying to figure out what parameters are driving the therapeutic outcomes. And in TENT-A1, we had different bimodal stimulation conditions, but all the arms had background noise. And we know that a lot of individuals use noise stimuli to help with their tinnitus, masking, or some residual effects. So we know it can be effective, but we also know that some individuals, don't necessarily like to include it in the bimodal stimulation. Now, at the heart of the bimodal stimulation, it's tongue stimulation paired with tones. That's the key. But we add in background noise that we believe can further enhance therapy, could also make it more comfortable to listen to. But we want to see, is that background noise a critical feature? And that was important. And so for our design, we had two arms. The first arm was, the clinical routine protocol that we use that you would see in clinics in Europe. And what we did with the second arm is that we removed that background noise. There's some subtle other differences, but that was the key difference. And we powered it to see if there was any significant difference between those arms, but also within arms, for each of those parameter settings, did you get significant improvements? And then, of course, 6 weeks into that, those two arms then got adjusted to a new stimulation setting because we want to answer that second objective. And in the paper, if people are willing to go there and want to learn more, we did some statistical compensation for these multiple comparisons, right, to demonstrate that it is significantly happening. And so overall, then, those two arms had the most number of participants. Of course, in a study, we can't look at everything, it'd be a 5,000 participant study, but we did add in two other arms, arm 3 and 4, that we can then look at exploratory questions. We can't say that they're significant because we didn't power for it or pre-specify, but we did design it so we can look at trends. And there we want to further see, we remove the background noise, but what if we remove the tongue component, or what if we add in more complex tones? We were able to look at that as well. And through the data, as we talk about it more, the key finding, not to give the punchline already, Neil, but the background noise is important, but it's not critical to get significant therapeutic effects. And the trends that we saw exploratory arms is that the tones and tongue are the critical features. And that's where we've converged on the study. So it's been very exciting to learn that. It's really about the tones combined with the tongue stimulation that's driving the significant therapeutic effects.
Brilliant. Okay, let's move maybe just to kind of show that in a slightly different detail, Hubert, which is the kind of timing of how people progress through the trial on our next slide. Oh, just before we get to that, I actually have a poll to run. I just wanted to check with our audience for 30 seconds, which of these questions best describe you. So there should be a poll coming up on your screen momentarily. Give Ross and Hubert a chance to catch their breath. before we get into the results discussion and really what it all means, we want to make sure we're tailoring the conversation to the audience. And so I'll just give about 10 or 15 seconds for the audience to answer any questions. Okay.
Are you going to be sharing the poll? Okay, yeah, it comes
yeah,
out.
so got the results on screen, and so predominantly people living with tinnitus and you know, some healthcare background. So we'll probably try and keep our discussion around the results, you know, as to a simplified manner, so our audience can digest them as best they can. Okay, let's move on. So in terms of the timeline then, so I suppose, you know, we obviously have, there's this screening and enrollment time that happens as per the graphic here on the right-hand side. And then the people go into a 12-week treatment period based on the treatment they're prescribed as per their group. So I'm kind of curious about why the 12-week treatment period was chosen. I think, Ross, you mentioned earlier, you know, there's this window of time from a neuroscience perspective that you know, to potentially, it's a good window to use. Is it that simple, or is it a bit more complex as to why we would choose 12 weeks?
Yeah, so the 12 weeks, as I said, yeah, it's that was a kind of a time frame that I suppose the field converged on. You know, it seemed to be the time needed to really kind of drive these neuroplastic processes and get hardwire these effects into the brain. So what we wanted to do with the TENT-A1 and also with the TENT-A2 studies was we wanted to see, you know, if you do intervene with bimodal neuromodulation for that 12 weeks and patients undergo this treatment regimen of 30 to 60 minutes per day over that time frame, and then if you stop treatment, you know, what happens thereafter? Do their tinnitus scores drift back to baseline? Does the therapeutic benefit that they've achieved within the 12 weeks, does that sustain? So it's an experimental protocol. And so the idea was to treat for 12 weeks and then follow up for 12 months. That was the design that we used in the TENT-A1 study. So as I said, one of the primary motivations for the TENT-A2 study was to replicate the results, to show that we could do it a second time. In addition to investigating all these other questions that that Hubert discussed, and that's really the motivation for the 12 weeks.
Very good. And I suppose, you know, following up out to 12 months after treatment in the 12-month follow-up period that's outlined here, you know, so they would have stopped treatment after 12 weeks in this trial. I think not to say that in the real world they can't continue to use it beyond that point, but just for the trial they stopped at 12 weeks. And I suppose, you know, with TENT-A, to be able to compare the results between TENT-A2 and TENT-A1, keep the same timeline, but probably important to gather any evidence about long-term effects of the treatment. Is that kind of the rationale for choosing the 12-month follow-up period?
Yeah, so we wanted to see, you know, how long does the effect of a 12-week treatment last? So I mean, we are breaking new ground here. Bimodal neuromodulation is a new field. You know, us as a company, you know, Hubert's lab, independently of us, before we joined forces, we're investigating it. You know, there's a couple of other labs around the world investigating it, but we're all of us together are making new discoveries. So the one of the motivations for this trial was, you know, if we give a 12-week treatment, how long does it last? And that's why we gave the treatment for 12 weeks. We took the devices back, and then we followed up with them, you know, 6 weeks, 6 months, and then 12 months to see, you know, how long do these effects sustain. And that, as I said, this was an experimental protocol, so it was designed to answer questions. Then in the real world, then that can tell us how do we actually deploy the treatment clinically. You know, we can, whether we advise patients to continue to use it beyond the 12 weeks, or is it that we just give them a 12-week treatment? So both of these trials helped kind of, I guess, elucidate that. But it's an ongoing process, and we will continue to research this area. As I said, we're breaking new ground. And, you know, as we continue our clinical research, the insights and findings that we find from our clinical trials and our investigations will then translate out into the clinical practices of the providers that provide our product in the real world.
Very good, very good. And we can see here kind of illustrated then that at the week 6 kind of interim visit, we were changing the stimulation or the treatment that they were receiving as described earlier. So, you know, it might have been synchronized tongue and sound for the first 6 weeks, and then changing maybe to a slight delay between the two to, I guess, stop people being used to the therapeutic effects, essentially. And we'll see the results of that in a moment, actually, when we move on to the next slide. But Hubert, maybe just before we get to the results, get to the, you know, the important stuff, how from this, how was the trial controlled? Because that's obviously an important question to ask when it comes to any clinical trial that's carried out. Maybe I can come to you to that for that, Hubert.
Yeah, definitely. So, you know, when you're designing a study, a controlled study, when people are referring to it is, you know, you have two arms, right? Two things you're comparing. You'll have your treatment, and then you have potentially some condition that is not the treatment, but ideally that the participants can't tell isn't the treatment. And that would be like a sham control. Sometimes people use the word placebo control. Our treatment approach, you can hear the sounds, you can feel the tongue stimulation. So we can't really tell individuals that they're not getting some, you know, that they are getting something when they're not getting it. So what's really, you know, one approach that's quite well accepted and is actually a quite rigorous way to do it is to have an active control. And the active control means that you actually have two of the treatment conditions, but you could think of them as, you know, two different doses or two different settings. It's like if you had, you know, a drug study and use different doses or kind of different treatments, right, for the same thing that you can't tell the difference. So that's the approach we did. And so this study was a randomized, blinded, because they can't tell, and a controlled study where arm 1 and arm 2, they were two arms, one active control, one that's, you know, arm 2, and arm 1 was what we use in the clinic. So that's the way we designed it. As we'll get to the results, we saw that both arms were effective. We didn't see significant between arms, but one of our pre-specified outcomes or endpoints was to show that you get significant changes within arm and that those are meaningful, right? That they do provide benefit. And so that's what we found, and we'll show the results that within arm, we saw significant changes, even more so than we saw in TENT-A1, which is quite encouraging. And that we saw that with or without the background noise, you're still driving significant improvements.
Very good. Okay. There's one question from someone in our audience, which I think is relevant just before we get into the results. You know, and the question is, when we spoke about neuroplasticity earlier, what is it kind of changing the brain to do? How is it retraining the brain? And I believe, and you know, please correct me, the idea is so that you pay less attention to the tinnitus, not that it's gone completely, but that you pay less attention, so it's easier to manage, less impact on your quality of life. Is that a fair simplification of the science?
Yeah, and you know, it is more involved, but just Neil, you can actually give the link to the TRI, Tinnitus Research Initiative Academy. I gave a talk there talking about the mechanism of action. There's already evidence, you know, anatomical, that there's connections between the tongue, the body, and the auditory system. There's physiological evidence that when you do that, you actually change the coding properties of these cells. You know, there's real changes physiologically happening in these cells where you're making them more sensitive to other things. And then it makes those cells less sensitive to other, you know, other sounds or features. And then there's also behavioral evidence as well. So all that, you know, I cover in that talk, so we don't need to go here, but you're actually changing the way those cells code to become much more sensitive to these other sounds and less sensitive to the tinnitus sound. And it's happening on a physiological and behavioral level.
Okay, very good. we will follow up with a lot of material links after the session for the audience as well. So I'm conscious of time, so let's move on to our final section, which is to discuss the results, and we'll keep a little bit of time for questions. So here are the top level results, which are impressive, I suppose. The first thing probably to clarify, which I think was touched on earlier, maybe by Hubert, is the measurement of these results are used using something called the Tinnitus Handicap Inventory, which is a clinical standard for measuring the effects of tinnitus, I suppose, on a given person. Is that a fair statement?
That's correct. And there's no objective measure yet of tinnitus. And in the end, what's important, and I'm a strong believer of this, is how the individual is doing in terms of their bothersome level of the tinnitus and their quality of life. And so the Tinnitus Handicap Inventory captures what we call tinnitus symptom severity. How much are they affected or bothered by the tinnitus? And so this tracks a score from 0 to 100. 100 being is the max, and then 0 being not bothered at all. And so that's what we're using for the study. And we use another one called Tinnitus Functional Index, but THI is our main outcome measure, and it is self-reported.
Very good. Okay, thank you, Hubert. So maybe we can compare then these results to TENT-A1, but before we do, we see 95% of people after 12 weeks report an improvement in this THI score, and 91% after 12 months. This is of treatment compliant participants. So compliance means they're using it as intended, I guess. Is what does that look like, Hubert?
Yeah, and this is where, me as a scientist, even though I am representing the company here, I like to be fully transparent and rigorous in the way we present the results. In the results paper, we do plot all the data for every participant that we have the data for, right? If individuals don't come back, we don't have that data, we can't present it. But those data are provided in the paper, and you can see, how each individual changes, and in the supplementary documents as well. So please, if you have any questions or interest, please go to those papers. What we're showing you here is 95% are those who use the device for at least 36 hours. That's what we pre-specified. It's based on previous studies that we found to be effective. We recommend 1 hour per day, but at least 36 hours in the 12 weeks is what we require. And when you do that, the individuals that came back and actually were able to do the tinnitus assessment, the THI, we found in all those individuals 95%, right? So it was, we're quite excited about those results. And then if you look at those who had improvements out to 12 months after the treatment stopped, it was 91%. And the other parameter you see here, well, actually stepping back a little before I get to that, there are questions of what happens to those who weren't treatment compliant and those who also didn't show up to do the assessments. And so if you were to say, and this is not necessarily true, but just to be fully, give the full spectrum, let's say all of those were non-responders, right? All of those individuals, many of them may have been responders and didn't show up, but let's say those were non-responders, meaning they didn't have an improvement in their THI score, then the percentage is 80%. 80% of individuals, they had improvements in their tinnitus symptoms based on the THI, which is still quite high considering the worst case scenario. So I just wanted to point that out. And then finally, the other piece that you saw there was, if you were to recommend the tinnitus treatment, and that was 88% that you saw listed there, which we are quite encouraged by, because that's just asking the individuals whether they improved or not, would you recommend it? So those are the results that you showed. And these are just getting more into the details where you can see there, 160. Again, as I mentioned, if you look at that plot on the left, 191. So there was about 31 individuals who either were not treatment compliant or didn't show up to do the assessments. But of the 160 that we had the data for, that's where the 95% comes from. But if you were to think of the worst case scenario where all those 31 individuals, were non-responders, that's where you get the 80%. So I just wanted to explain that so individuals, if they're reading the papers, can fully understand the full range of what individuals improved in their scores or not. Very
good. So I think to your point, Hubert, these these two graphs show the individual results of each of the participants we had the data for 12 weeks of treatment and 12 months after the treatment ended. And is it fair to say then that anybody to the right of the line will be classed as someone who had any improvement in their symptoms? Is that accurate?
Exactly. And just for those who are going to read the paper later, the x-axis there tells you what their THI score was before the treatment, right? And then the y-axis is showing you what their THI score after the treatment at 12 weeks. And so if you have a higher score before the treatment and have a lower score after the treatment, then you're going to fall on the right side of that diagonal line. And each point is an individual, right? Is a participant in the study. So this is what was quite astonishing compared even to TENT-A1, but other studies is nearly all the points are below the diagonal line, right? And that's what we mean by they've had an improvement in their THI score. We'll get to it later because you may wonder, what do these changes in THI score mean? Another way to look at the data is just to ask individuals who were in the study, did you benefit? And we did ask that as well. And things were quite consistent with all these values and percentages that we're presenting.
Very good. Okay. So let's compare, I suppose, these results to TENT-A1, because part of the reason the trial was carried out was to see, if we could, as Ross mentioned, replicate the effectiveness and see if changing the stimulation received after 6 weeks led to further improvement. So I suppose what we can see here is maybe just a little bit of background for people viewing, is the descending lines show an improvement or a reduction in the THI score, which should correlate to an improvement in quality of life. And the deep blue, the deep purple is TENT-A1. And what we see is after the 6 week point, TENT-A2 does seem to further improve or further decrease the THI score. So is it as simple to say maybe that changing the stimulations seem to work from the trial's perspective? Is it that simple to state an outcome, or is there something more at play?
Yes, you can. And we did design the study to look at that question. And if you look at the results paper, there's something called Bonferroni correction, which is, I won't get into the jargon, the details there, but you do have to account for doing multiple tests and so forth. And what we showed was that you did get a significant improvement for the second 6 weeks, when you change the stimulation parameter, and we didn't see that effect in TENT-A1. And you can actually see from here that it plateaus, right? There's a plateau effect. So we did get significant improvements in that second 6 weeks. And just to give you some numbers on average, it was about 14 points compared to about 19, 20 points, depending on which arm. And so we were able to get almost 3 times what people define as the clinically significant or difference of 7 points. So you want to get 7 points. We were almost 3 times that in TENT-A2 and about double that in TENT-A1. So we were quite excited about that further improvement. And you can see in both cases, you get a sustained effect out to 12 months.
Great. So I suppose the clinically important number of 7 points, that should correlate to improved quality of life. Is that fair?
Correct.
Interesting. Okay. So I'm interested, Hubert, we the results look very strong, obviously, but in some of the paper, I think it's 79 participants in total reported that they had, an increase in their tinnitus or a fluctuation in their tinnitus, I believe. So how can that be true of 79 people reporting that during the trial? How can that be true, but also these results be true? Are they mutually exclusive, or how can we reconcile those numbers?
Yeah, so I also saw a few questions asking about, was there an increase in tinnitus or fluctuations and so forth from the device? Many of you who have tinnitus on this webinar know that there is some fluctuating aspects of the tinnitus, and some days it can increase, some days it can decrease. And so it's sometimes hard to tell what is driving those changes. And other times you can have increase in tinnitus, but they're not necessarily bothersome. and they could be short-lived, right, transient. So we do report every incident that we see if an individual has an increase in tinnitus. And so that's where the 79 comes from. Just to further break it down though, of those 79 increase, we could only say that 7 were, really we're sure, well, not sure, but, we can claim as related to the treatment out of those 79. The rest we had to label as possibly or probably, and these are just that there's not enough information to make a, causal link. But with that said, then you have the tinnitus that can increase, but it may not actually be bothersome to an individual. It may just be a change. It's reporting the change. And then also it eventually can drop down. And we do know that in the early parts of the treatment, people are reporting that they get changes in their tinnitus, which is a good sign because it means that things are changing in the brain, right, to the treatment. And then by the end of the 6 to 12 weeks, they are actually experiencing improvements in their tinnitus symptoms. And so that's why the numbers may show 79 increase in tinnitus, but it doesn't necessarily mean that they're not benefiting from the treatment, which we would look at more the tinnitus symptom scores with THI. And also, as you'll show in the later slide, we just ask individuals, did you benefit, regardless of fluctuations or not? And that to me is a powerful indicator.
So it's fair to say that we're just doing our due diligence on our reporting when there's 79 people, noted in the trial as having reported an increase in tinnitus, but it in the vast majority, or maybe all cases, it didn't persist throughout the treatment, because obviously the overall reporting of the results are quite positive, I suppose.
Exactly, and only 7, I'll reiterate, were causally related out of the 79.
Very good. And I suppose, Ross, it's obviously quite significant, taking a step back to replicate the results of TENT-A1 or further improve generally, but specifically for 12, out to 12 months. That must be something that's quite confidence building, I suppose, with further research or clinical protocols in the real world.
Yeah, so as I said, one of our motivations, when we looked at the clinical need, was, as I said, sound therapies and hearing aids. They're effective while in use, but when patients take them off, then there's no residual effect, and their patient's tinnitus comes back straight away, and often actually spikes and gets a lot worse. So what we were trying to do was trying to find a sustained, kind of sound therapy effect. So that's what our data is pointing towards. We've got here, so it is very significant. So we're very excited about it. I think it's what the market has been asking for. So it's what we're trying to develop, and it's something that we'll continue to pursue. And we built on the success of the TENT-A1 study. We got further improvement in the TENT-A2 study. And as I said, as a company, we're committed to clinical research and to continuing to improve and build on these early successes, and just keep making our technology more and more effective as we answer more of the questions as we break new ground here.
Very good. Okay, so we just have maybe two final points before we get to some quick questions, because I'm conscious of time. So we have a comparison here between TENT-A2 and TENT-A1, which just shows the maybe increased numbers of people who reported a benefit after 12 weeks of treatment in TENT-A2 to TENT-A1, and we can send those materials on after today's webinar. But I just wanted to touch on something, Hubert, before we wrap up, about, you mentioned asking people how they felt, or if they felt they benefited at the end of the trial, or at the end of the treatment, excuse me. So maybe you can just add a bit of color commentary to that and what we asked them.
Yeah, and to me, this is the most telling, right? Because those THI are important for rigor and clinical trial design. But to me, I mean, you just asked the individual, did you benefit or not? I mean, that's really what matters. And also, would you recommend it? So these are two questions I think are critical, and we asked in both studies. And you could see on the right side for TENT-A1, did you benefit, from here I have to look. It's on the they're both stacked up on each other here. But for TENT-A2 on top, you'll see that they had yes was 80. Is this mixed up here? Sorry. Oh yeah, would you benefit? They're on the bottom. I apologize. They're on the bottom there. So if you look there on TENT-A1, you have 66% said yes, and then you had 70% say yes in TENT-A2. So there was a bump increase there. And then if you look at TENT-A1, would you recommend this treatment? You had 77%, and in TENT-A2 you had 87%. So this is, forget about THI and the scores and so forth, and what do they mean? If you just directly ask them, you're getting about 2/3 benefiting and close to about 80 to 90% recommending the treatment. So I think that's important to understand in the end. If people are really trying to understand, how did it affect them truly, this is what I think is the most important result.
Okay, very good. That's a good takeaway. So we're just about out of time, so I'll move beyond our final discussion point, which is more data and results heavy, and we might just get to a few quick questions for a couple of minutes before we wrap up. We will be distributing a recording of the webinar after the fact, and we will answer any questions to those who asked over the next couple of days if we don't get to it now. But we have a little bit of time here just to answer for those who've stayed on throughout the session. So the first one, Ross, maybe this is a question for you, is in relation to US availability and if there's any update on that front.
Yeah, so we're currently working with the FDA towards getting Lenire approved in the US. But at this time, I don't have an update. But if patients want to follow us either on various social media, or if they want to sign up for updates, we can let them know as developments come along.
Very good. In terms of Hubert, maybe I might come to you for this next one. Someone asks here, is the effectiveness of bimodal neuromodulation adversely affected if it's used in combination with other therapies, maybe such as CBT, for example?
The answer is, we don't expect so. In fact, it should complement and supplement these other approaches. If you think about what we're trying to do, which is, reduce the bothersome nature and awareness to the tinnitus sound. And if you think about what CBT is doing, or these other kind of counseling psychotherapy, they're also trying to do that, but on a cognitive level. We're trying to do that more behind the background, automatic, to enable them to do that. So I do think you could combine these different approaches and further enhance the effects. Even if you were to try different sound therapies mixed in, that's what we're doing in essence with our auditory. We can think about using other types of sounds and combining with bimodal, or even with hearing aids with tongue stimulation, right? So these are all options that I think have potential in the future.
Very good. Quick question here is, can the device be used for people who have tinnitus but also have hyperacusis?
So I can answer that as well. Or Ross, go ahead. You could do this.
Yeah. I was going to jump in. So now we haven't published the subtyping data yet. We are going to publish that, I believe. But as I said, the TENT-A1 and TENT-A2 studies, they looked at two kind of areas. One was the differential effects that different stimulation parameters had on the overall population, but then also what differential effects they might drive in patient subtypes, such as patients with hyperacusis and tinnitus. So we actually found that patients with hyperacusis were very responsive to our synchronous high frequency stimulation. So they are a group that appear to be particularly responsive to the treatment. So yeah, I think that if a patient has hyperacusis and tinnitus, I think it would be a very good idea for them to try our technology, because they keep popping out as a subgroup that seem to do very well with our technology. But as I believe that's in the publication plan, Hubert and his team are going to publish the subtyping data from TENT-A2, and so they'll be able to read that when it's published.
Very good. Maybe just a couple, maybe two more before we wrap up. There's a lot of questions, but we'll get to them in the coming days, as I said, if we can't get to them now. So someone here asks, can the device help with no longer being bothered by tinnitus at night, and is there any advantage to continuing treatment after the 12 weeks? Maybe Hubert, I might come to you for that one.
Yeah, I see that question there. They're asking, they did 2 10-week treatment sessions and they're no longer bothered at night. The individual mentioned this and asked if continuing to use the treatment could be advantageous. I will say, we in our data, we do show that there is long-term effects after 12 months, and that was encouraging. However, full transparency too, the data, even if you look at our 10-A1 paper in Science Translational Medicine, you do start to see eventually the tinnitus benefits are starting to fade away, right? And this is not surprising because we know in the brain, we're not curing tinnitus. So if you're not tackling the source, then whatever the source is still there and it's going to drive it. So how fast you need to keep continuing, to the person who asked this question, we don't know that answer. We don't have the data yet, but I would suggest not to completely stop treatment. I would do it periodically, especially if you do notice the tinnitus symptoms are getting, coming back again. But that is very encouraging that you're no longer bothered at night. So that's great to hear.
Yeah, and I think in addition to that, I would say that, as I said when we were discussing the clinical trial, that the 12-week treatment protocol was an experimental protocol. It was, that driven by that neuroplasticity window. But in the real world, patients are using it more in a chronic kind of care model. Now they seem to be using it in one of two ways. They either, as Hubert said, they use it to get better. They take a break from using it. Then if they feel that the tinnitus is coming back, they almost top up with another treatment, or they continue to use it. So that is probably, it's great. It's fantastic to hear these success stories, particularly the patients are sleeping, because I know from my experience of speaking to patients, if you're losing sleep, it's even small problems become a very big problem. As you are losing sleep over nights and nights and nights. So it's great to hear that it's having that benefit. But I would say to that patient, it's if they feel like that the symptoms are coming back, then possibly top up with another treatment, because that is what we're seeing in our real world data as well.
Very good. Okay. Just a couple of final ones before we close. We've nearly 100 questions in, so hard to get to all. Hubert, we spoke about control earlier, but is it possible that the results are impacted by a placebo effect of any kind?
So there's always going to be some placebo effect. Just to be fully transparent and straightforward with that. But there's two things to keep in mind. One is that we did see this 12 month effect lasting, and for me, even considering to be very critical, it's hard to imagine that would be a full placebo effect. You stop the treatment at 12 weeks and you keep going for 12 months. So the treatment could have some placebo component, but can't be fully due to placebo. The other thing I mentioned earlier, which is quite exciting with the results, is that we changed the stimulation parameters midway, and for arm 1, the individuals can't perceive that change in the stimulation parameter in the setting. It's just changing delays. And even in clinic, individuals always ask, did you actually change my stimulation setting? So the fact that you could actually drive another 30 to 40, 50% more improvement without them even perceiving that change is also further evidence that it can't just be placebo alone. So these are different factors which we're quite excited about, that drive to being able to drive symptom improvement in symptoms. That's not just solely due to placebo.
So I suppose placebo by definition means you think you're using something that isn't actually providing an effect. But if the treatment stopped at 12 weeks and they're followed up with a year later when no one's been using anything, by definition, that 12 months data point can't really be placebo, because there's nothing they think they're using. Is that a fair summarization?
Very good point. Yeah, good job, Neil. Yeah, exactly. That's the point too. And if they can't perceive something changed, then it's hard to imagine that they're having a placebo effect to something that they didn't know changed, right? So that's another example.
Also, I'd add to that some of our colleagues in the tinnitus research field, like Professor Berthel and Guth and others, have characterized the placebo effects that they observe in some of their publications. So they'll often say that, you might see a short-term placebo effect, but then patients drift back to baseline and often sometimes get worse. When the placebo effects wear off and they realize that the intervention is not having an actual therapeutic effect. So at this point, we're completely confident that what we're seeing here is a placebo effect.
So look, we are over time. A lot of people have stayed with us, and we've a lot of questions to answer, but those will be followed up with in the coming days, along with the recording and some additional materials that we mentioned throughout in terms of research papers and other literature, which will be sent to everybody who registered. All that's left for me to do is to thank Hubert and Ross for being so gracious with their time and answering the questions in a way I hope that's beneficial to everybody who joined us this evening, or wherever you are around the world, if it's morning somewhere, and to thank you for taking the time to listen to the results of TENT-A2 and the second large-scale clinical trial of Lenire. Lenire is available in Europe. You can go to lenire.com for more information about that, and as Ross mentioned, currently engaged with the FDA in the US. So that's it for this evening, for this session. Thank you very much, and we shall see you again soon. All the best.